Okay, the point of this video is to give some training on the diversity and taxonomy of the Ralstonia solanacearum species complex, which cause wilt disease of plants. I am Professor Tiffany Lowe-Power at UC Davis, and I've been working on Ralstonia for over a decade now. So first, I want to go into some of the taxonomy and genetic diversity of these pathogens. Then I will summarize our group's work to conduct a literature meta-analysis of all of the isolation reports of these species and our goals of visualizing these as a dashboard. And then in a second video, I will show some examples of how to use that dashboard to answer questions about which strains are where, and what patterns of hosts of isolation are present in different lineages, and other epidemiologically relevant questions. Okay, so this is a slide that shows some of the taxonomy and nomenclature in the Ralstonia solanacearum species complex. So these organisms are a species complex, which means that they are more diverse than a single species of bacteria, but functionally similar enough that it makes sense to refer to them under one semi-informal name of a species complex. There are four phylotypes of Ralstonia, which I'll get to and explain on the tree to our left. And as of 2014, these organisms have been separated into three species, with that initial species separation being proposed in a 2011 genomics paper. So the three species in the species complex are Ralstonia solanacearum, Ralstonia pseudosolanacearum, and Ralstonia syzygiae. And what I'm showing on the left of this slide is a phylogenetic tree that represents the species diversity of these strains. Each leaf on this tree is a publicly available genome of a Ralstonia solanacearum species complex organism. And in color, I have color-coded the different phylotypes. In several cases, the phylotypes directly correspond to one of the now recognized species. So Ralstonia solanacearum is phylotype 2. Additionally, phylotype 4 directly corresponds to the species Ralstonia syzygiae. But there is one case where there are two phylotypes, phylotype 1 and phylotype 3, that are close enough related to each other that they are recognized as being in the same species of Ralstonia pseudosolanacearum. And you can see that that is represented on this tree where they are more closely related than these other groups, which are more diverse. It's always been a bit of a sad thing in history for me that phylotypes 1 and 3 happen to be the most closely related, but that is the order in which they were named in the early Ralstonia genetics papers. So, okay. In addition to having these phylotypes and species, there is significant diversity of different groups of organisms in the species complex that have been called sequence variants or sequevars. The sequevars 1 and sequevars 2 correspond to the pandemic lineage of potato brown rot. This sequevar, so there's a B designation in phylotype 2. And so these 2B1, 2B2 strains are what we also know as race 3 Biovar 2, Ralstonia solanacearum. So this is a clonal lineage, which you can see on the tree here, this flat branch, that has been distributed to many countries beyond the original origin of this species, or origin of this lineage. So the pandemic lineage originated in the Andes Mountains and has were co-evolved with potato and through human movement of plant products, this lineage has been introduced and become established in many countries around the world. And so it took some time for the genomics to catch up to the historical literature on taxonomy. But the current recognition is that what has been known for nearly half a century as race 3 Biovar 2 corresponds to this 2B1, 2B2 clonal pandemic lineage. And that is nested within Ralstonia solanacearum. One hurdle in taxonomy is sometimes names get proposed and start to be used. And so there has been a recent proposal to divide one of the species into two species. So the proposal is to divide phylotype 1 into a novel species that was named by these authors as Ralstonia nicotianae. And then that would mean that in their proposal, phylotype 3 would be the only strains that are known as Ralstonia pseudosolanacearum. Unfortunately, it's the opinion of many researchers in the Ralstonia field that this hasty taxonomic proposal was based on flawed data and proposed an inappropriate cutoff to name a different species. If you're interested in the details of that, we have this open access rebuttal. Regardless of where you feel like the division is correct, I want to bring this up because unfortunately some of the databases have started to pick up this flawed name of Ralstonia nicotianae. And so it's worth everyone knowing that this is actually phylotype 1 in Ralstonia pseudosolanacearum. So this group, to go back to that tree, all of these yellow strains are in phylotype 1, and that is what was proposed to be moved into this new species. Um, so. Without going into all the details, I wanted to bring this to your attention. Okay, so one of our goals has been to try to understand what is known about the diversity of these organisms. And one of the ways that we wanted to go about this is by understanding the literature. So Ralstonia, because it's a major pathogen, has been studied in many countries in hundreds and hundreds of papers that have looked at surveys or other epidemiological analyses to analyze different strains of Ralstonia, where they've been isolated, what plant they've been isolated from, and sometimes their genetic identity within the species complex. And so for several years now, our lab has run a meta-analysis project to consolidate all of this information into a large Excel spreadsheet. And we've been releasing this Excel spreadsheet as what we call a living preprint on bioRxiv, where we're updating the supplemental table as we have different releases and additional information that we bring into it. So this is what that Excel spreadsheet looks like. Wherever possible, we record the taxonomy and phylogenetic information, so the phylotype and sequevar, strain name when it's listed. We record the host plant of isolation at multiple taxonomic levels, year isolated, the location isolated at multiple geographic levels. When it exists, we record sequence successions for either the marker gene, EGL is what is commonly used for those sequevar assignments, or whole genome accessions, which are becoming increasingly common. And then also a reference where that citation came from. And as of fall 2025, information from almost 10,000 Ralstonia solanacearum species complex strains has been recorded from over 105 locations, which are typically referring to countries here, but sometimes geographically separated territories. Yes. And so, although what I'm about to show later is a dashboard that summarizes some of these patterns, there might also be folks who are interested in curating and combing through this Excel spreadsheet. So it has been available on the bioarchive and the supplemental material. And anyone can sign up for updates to follow this preprint to get automatic emails when we post a new version of the data set with additional information. And so what we have is this issue where we have all of this metadata on these strains, but that's a lot for any individual person to comb through. And so our solution has been to build an interactive dashboard. We used our Shiny code and a really nice example on the Fusarium graminearum species complex from Emerson Del Ponte in Brazil. He was kind enough to share his code, which we adapted into this dashboard. And so in a second video, I'm going to record some multiple case studies into how to use this Ralstonia diversity dashboard in order to answer different epidemiological questions about Ralstonia as gleaned from published data. Of course, this is a project that's based on published data, so it is not necessarily complete to the full biological picture. Of course, there will be sampling biases in any individual study. There is disparity in the amount of funding for Ralstonia research in different locations. And so the abundance and trustworthiness of the data is going to vary across different studies. So the global picture should be used to get an understanding, but not used with high certainty for areas where decisions need to be very careful. Okay, so I will now pause and end this recording, and there should be a link to the next recording under this video. Additionally, here's the full URL that you can type out. If you're allowed to use shorter URLs and prefer to type out the shorter one, this is a redirect that goes to that same dashboard. It takes a few minutes to load, maybe up to a minute. It basically processes the code on a remote dashboard or a remote server each time you use it. So have a little bit of patience, but once the initial loading happens, it does get a little faster.